Dear students, this is Dr. Shabana Begum, Associate Professor in the Department of Zoology, Maharani Science College for Women, Bangalore, presenting a lecture on body coelom, a subtopic of Unit 1, that is Animal Architecture, pertaining to Zoology, first semester. Assuming that you have understood the previous lectures of mine on the definition and types of body symmetry, body organization and germ layers, including diploblastic and triploblastic conditions in animals, let me explain about the subtopic body cavity today. So the learning objectives of this topic being to define body coelom and study different types of coelom, and to understand evolution of body plans in animals. The three figures A, B and C in this slide will give you the diagrammatic sectional view of coelomate, pseudocoelomate and acoelomate condition respectively. So coming back to what coelom is, first let us give a small introduction about it. The term coelom was suggested by Haeckel in the year 1872, and the term is derived from the ancient Greek word koilia, meaning cavity. So the coelom is defined as a fluid-filled perivisceral cavity lined by epithelial cells derived from the embryonic mesoderm. So, coelom is the main body cavity in most animals and is positioned inside the body to surround and contain the digestive tract and other organs. In some animals, it is lined with mesothelium, while in others, such as molluscans, it remains undifferentiated. In the past and for practical purposes, coelom characteristics have been used to classify bilaterian animal phyla into informal groups. So this coelom cushions internal organs, allows internal organs to move independently of the outer body wall. Then it acts as a hydroskeleton in some animals such as earthworms. So types of animals based on presence or absence of coelom. So depending on which the animals are classified as the acoelomates, that is the animals with no coelom; coelomates, the animals with a true coelom, that is the coelom here forms from the mesoderm to become mesenteries and suspend internal organs, and the pseudocoelomates are the animals which form a cavity from the blastocoel. So you can maybe elaborate on the three types of coelom once again. So among the lowest bilaterians, that is triploblastic forms, the space between the body wall and gut is filled up by parenchymatous cells derived from the mesoderm. Such animals are known as acoelomates. Examples are the forms belonging to phylum Platyhelminthes and Nemertini. In some bilaterians, the space between the body wall and gut encloses a fluid-filled cavity which is not lined by mesodermal epithelial cells, and such a coelom is known as the pseudocoel or false coelom, and the animals possessing it are known as pseudocoelomates. Embryologically, the pseudocoelom is derived from the blastocoel of the embryo. The internal organs are free within the space as there is no peritoneum bounding the cavity. The examples are the forms belonging to the phylum Nematoda, Acanthocephala, Entopracta, Rotifera and Gastrotricha. While in rest of the bilaterians, the body cavity present between the body wall and the gut is lined by a peritoneum, which is formed from the embryonic mesoderm, and this type of cavity is known as the true coelom, and the animals possessing it are known as eucoelomates. Fine. You can mention the outer covering of the coelom is called the somatic or parietal epithelium, and the inner layer is called the visceral or splanchnic layer or epithelium. In this coelom actually contains most of the visceral organs. So the mesodermal lining or peritoneum allows organs to be attached to each other so that they can be suspended in a particular order while still being able to move freely within the cavity. There are three subtypes of coelom, such as schizocoelom, hemocoelom and enterocoelom. In these figures, the first row of diagrams in the slide will depict the development of schizocoelous coelom. Here, the schizocoelom actually develops from the split in mesoderm and is found in annelids, arthropods and mollusks. While the second row of figures in this slide will depict the development of enterocoelom, that is, it develops from wall of embryonic gut or enteron as a hollow outgrowth and is found animals from echinodermata to chordata. While the hemocoel actually is a true coelom reduced and the body cavity here is filled with blood or hemocoelomic fluid which contains plasma, amoebocytes and hemoglobin circulating throughout the body and functions like blood. The examples being the phylum forms belonging to the arthropoda and the molluscans. So more about enterocoelom and schizocoelom. So schizocoely is exhibited by forms called protostomes in which the blastopore, that is the opening of archenteron during gastrulation, forms the mouth aperture, while in case of deuterostome the coelom is called enterocoely, where actually in case of deuterostomes the fate of blastopore is the formation of anus. So from blastopore anus is developed during development. In case of schizocoely or protostomes which exhibit it, the archenteron initially forms and the mesoderm splits into two layers. The first attaches to the body wall or ectoderm, forming the parietal layer, and the second surrounds the endoderm or alimentary canal, forming the visceral layer. And the space between these two layers is known as the coelom or body cavity. So in case of deuterostomes, the coelom forms is by enterocoely. The archenteron wall produces buds of mesoderm and these mesodermal diverticula hollow to become the coelomic cavities. So deuterostomes are therefore known as enterocoelomates. Examples are cited here. You please go through the slide later. Coming back to the origin and development of the coelom. So the evolutionary origin is uncertain, and the oldest known animal to have had a body cavity was the Vern animalcula. Currently, few hypotheses or theories have been put forward, but only 4 of them are of importance, which are as follows. The first being the acoelomate theory is also called schizocoel theory, which states that coelom evolved from an acoelomate ancestor like flatworms. And according to this theory, acoelomate body plan is primary and ancestral to the coelomate plan. Second theory is called enterocoel theory. It's put forth by Lancaster in the year 1875, which states that the coelom evolved from gastric pouches of cnidarian ancestors. This is supported by research on flatworms and small worms recently discovered in marine fauna. The third theory is called gonocoel theory, proposed by Berg in the year 1885, which states that coelom evolved from expanded gonad. This theory has no embryological support because gonads do not arise before the coelom. There is one more theory called nephrocoel theory, proposed again by Lancaster in the year 1874, which states that coelom evolved as an expanded nephridium. So no supporters to this theory as protonephridia are seen in coelomates, while excretory organs are absent in some coelomates like echinoderms. So coming back to the functions of the coelom, a coelom actually absorb shock or provide a hydrostatic skeleton to assist in locomotion and maintaining body shape. It can also support an immune system in the form of coelomocytes that may either be attached to the wall of the coelom or may float about in it freely. The coelom allows muscles to grow independently of the body wall. This feature can be seen in the digestive tract of tardigrades, that is the water base, which is suspended within the body in the mesentery derived from a mesoderm-lined coelom. Some more points of advantages to have a body cavity. That means the coelom provides more room for organ development, more surface area for diffusion of gases, nutrients and waste into and out of organs. It acts as an area for storage of gametes. Then it acts as a hydrostatic skeleton. Then it forms a way to eliminate the waste products, and also it facilitates increased body size. And it acts as a site for gamete maturation and brooding of embryos. In some animals, it keeps the body wall moist to facilitate respiration and to destroy bacteria and other harmful microorganisms. To learn more about coelomic fluid, so it's a fluid inside the coelom is known as coelomic fluid, which is a colorless watery alkaline fluid containing many salts, proteins and amoebocytes called coelomocytes. This fluid is circulated by mesothelial cilia or by contraction of muscles in the body wall. In case of annelids, the coelom is divided into fluid-filled compartments by transverse partition called septa, which extend from the annuli of the body wall to the alimentary canal. The wall of the septa is perforated, through which coelomic fluid communicates from one compartment to another. The coelom communicates with the exterior by two sets of ducts, namely nephridia and coelomoduct. Nephridia are ectodermal tubes that remove water and excretory waste, while coelomoducts are mesodermal tubes which usually open into the coelom at one end, while the other end communicates with the exterior. Some functions of the coelomic fluid, which are similar to the functions of coelom. Coelomic fluid acts as a hydroskeleton. It allows free movement and growth of internal organs. It serves for transport of gases, nutrients and waste products around the body. It allows storage of sperms and eggs during maturation, and it acts as a reservoir for waste. So this slide will figure out the triploblastic body plans in acoelomate, pseudocoelomate and coelomate organisms respectively. So in case of acoelomates, here you can see there is an outer layer of ectoderm. This is a sectional view. Then the middle pink cells, it's a solid mass of tissue it forms, forming the mesoderm, and the yellowish cells form the endoderm, and there is a centrally placed digestive cavity. While in case of pseudocoelomate conditions seen in case of roundworms, you can find here the outer covering of ectoderm, middle mesoderm and the inner yellow colored cells of endoderm. But as such, here the coelom is not lined by the epithelium derived from mesoderm. Hence, it is a pseudocoel or false coelom. In case of coelomates such as mollusks, annelids, etc., the condition is little different. Here again you can find in the section the outer ectoderm, middle layer of mesoderm and inner endoderm. Here you can see the schizocelic development of the coelom. You can find here there is a split in the mesoderm layer to form the coelom or body cavity, which appears whitish in color. So a true coelom is derived from mesoderm, and its significance being a true coelom is lined on all sides by mesoderm, which gives rise to muscles that surround the gut as well as underlying the body wall. This allows more efficient digestion because food can be pushed through the digestive tract by muscles. The examples of coelomate, here the earthworm and its section is given in this particular slide. The second diagram of roundworm, which is a pseudocoelomate, along with its section, is also depicted in this slide, where a pseudocoelom is a body cavity derived from blastocele rather than from mesoderm. Finally, in the third diagram, a planarian is shown along with its section. Here these are the organisms without body cavities and are considered acoelomates, and here the relatively solid mass of cells are seen between ectoderm and endoderm. So coelom is absent. Some more points regarding acoelomate phyla. Acoelomates lack a fluid-filled body cavity between the body wall and digestive tract. This can cause some serious disadvantages. This is the disadvantage to be an acoelomate here. The fluid compression is negligible, while the tissue surrounding the organs of these animals will compress. Therefore, acoelomate organs are not protected from crushing forces applied to the animal's outer surface. The examples of the acoelomate phyla already mentioned in the previous slides. You can just go through these examples once again. Coelomate phyla, according to Brusca and Brusca, are the nemerteans, annelids, arthropods, mollusks, then hemichordates, chordates, etc. Finally, the pseudocoelomate phyla are the rotiferans, nematodes, then acanthocephalans, lanciferans, etc. So these you can go through and learn little better. So in a layman's term or language, acoelomate organisms are without body cavity, examples being the platyhelminth swarms. Pseudocoelomates are with false coelom, examples Ascalminthus, and the coelomates are with true coelom, and the examples are the annelids, arthropods, mollusks, echinoderms, hemichordates and chordates. So the outcome of the study, that is what have you studied in this particular topic, in a nutshell, to say presence or absence of a coelom or a cavity between the body wall and the gut wall is very important in classification of organisms. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. Examples are annelids, mollusks, hemichordates, chordates, etc. And in some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between ectoderm and endoderm. Such a body cavity is called pseudocoelom, and animals possessing them are called pseudocoelomates. For example, the Ascalminthus swarm, that is roundworms, etc. The animals in which the body cavity is absent altogether are called acoelomates, the example being the cnidarians and the platyhelminthus forms. Once again, I can say the schizocoelom develops from split in mesoderm cell mass, which arises from the walls of the archenteron. Schizocoeloms are found in animals which are known as protostomates. Then enterocoelom develops from the wall of embryonic gut in the form of coelomic pouches, which are the invagination of the endoderm of the archenteron. Then enterocoeloms are found in animals such as deuterostomates. So with this, I have concluded my lecture, and now let us discuss some multiple choice questions belonging to this particular lesson. The first question: the body of which diploblastic animals are derived from only two embryonic cell layers, that is ectoderm, endoderm, but no mesoderm? The answer options are sponges and coelentrates, protozoans, microbes, and none. The answer option A, sponges and coelentrates, is the correct one. Second question: acoelomates are characterized by the absence of brain, absence of mesoderm, deuterostome development, solid body without a cavity, surrounding internal organs. The option D is the correct answer. Third question: The blastopore denotes the presence of an endoderm-lined cavity in the developing embryo, a cavity that is known as the options are archenteron, blastula, coelom and germ layer. The answer being the option A, archenteron. Fourth question, the blastopore is a structure that first becomes evident during. Options are fertilization, gastrulation, 8-cell stage of embryo and cleavage. The answer is the option B, that is gastrulation. Final question, the organisms that are neither coelomate nor pseudocoelomate should, apart from their digestive systems, have bodies that, options are solid with tissue, lack the ability to metabolize food, lack true tissues, lack mesodermally derived tissues. Answers are the options A, that is they are solid with tissue. Finally, these are some of the references for you to just go through or learn once again if you have any doubts regarding the topic. Thank you, girls.